Okay, perfect. Now, let's understand the arrangement of a centriole. So, as of yet, what we studied was that we have tubulin proteins. Those tubulin proteins are of two types, alpha and beta. They come together to form a tubulin dimer, and those dimers polymerize to form protofilament, and 13 protofilaments are making up one microtubule. ठीक है जी? Now, what happens is that we have lots of microtubules present in our cell. Say yeah. Now, microtubule is again a cylindrical structure, something like this. So let's see. So over here, I have drawn three microtubules. These are three microtubules. Can I say that this structure is basically a microtubule triplet? Why? Because three microtubules are attached to one another, they're connected to one another, and I can call this structure as a microtubule triplet. ठीक है जी? Now what happens is that when 9 microtubule triplets wound in a hollow cylindrical structure, and what do I mean by that? Let me draw a cylinder. ठीक है? This is a cylinder, and now can I say this is one triplet? And then I draw another triplet. 1, 2, 3. And then there's another triplet. 4. And then 1, 2, 3. We have our fifth triplet. Then we have our sixth, seventh, eighth, and ninth triplet. And obviously, दूसरी side पर भी इस तरह से run कर रहे होंगे in the form of chain. 3 microtubules are 39 protofilaments. Yes, so basically, we have 13 protofilaments over here. We have 13 protofilaments and we have 13 protofilaments, right? So we can say that a triplet has 39 protofilaments. You're absolutely right. So let's see over here. We have drawn 9 microtubule triplets, which means how many microtubules do we have? 1, 2, 3, 4, 5, 6, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26. This should be 27. What have I missed? 1, 2, 3, 4, 5, 6. Okay, I have written 6 twice over here. My bad. So basically, all of this would add up to 27. So let me just fix this. So basically, 9 microtubules triplets mean that we have 27 microtubules present. So I can say that when 27 microtubules join together in as 9 triplets, we have 9 triplets, and these 9 triplets are forming a hollow cylinder. And basically, ये जो पूरा region होता है, this entire region is a hollow cylinder. And what do I mean by hollow? If you look at a pipe and if it's an open pipe, यानी आप उसके दोनों ends को देख सकते हो. So basically that pipe is a cylinder, and if you can see through both the ends of that pipe is hollow. There's nothing inside the pipe, right? So that hollow pipe, that hollow cylinder, we say that it's an open cylinder, right? So over here, our centriole is basically, the arrangement of a centriole is called a 9+0 arrangement. Why? Because we mean that we have 9 microtubule triplets and there is a hollow center. यानी जो surrounding है उस cylinder की, the boundary of that cylinder, this entire boundary of that cylinder, it has 9 triplets. There are 9 triplets present on the boundary of that cylinder, and from within, from the inside, from the center, it is hollow. And this arrangement is referred to as 9 plus 0 arrangement. And your centrioles are always and always present in a 9 plus 0 arrangement of microtubules. Is the structure of centriole clear to you? Any questions? So over here, this is exactly how a centriole look like. So let's see. We have, this is one triplet. So this is one triplet. This is second, third, fourth, fifth, sixth, seventh, eighth, and ninth. Okay, so these are basically our 9 triplets over here. And these 9 triplets are basically forming what? They're forming this boundary of our centriole. And from the center, from the middle, this is basically a hollow body. And obviously these microtubule triplets are connected by little small fibers. So this is how a centriole looks like. And obviously इसके right angle पे एक इसी का similar centriole present होगा, because the centrioles are always present as pairs. So yeah, that is centriole for you. The function of a centriole is basically in cell division. Function of a centriole is in cell division, and that too in the formation of spindle fiber. Okay, that's all for centriole. Any questions? Or else I will move to the structure of cilia. Any questions? Everything is clear? Okay, so now that we've studied centriole, let's look at something. Now, cilia are what? Cilia are basically hair-like projections, hair-like strands, thread-like strands. They're very small. They're like small hair protrusions coming out of a cell. So they're always present on the cell surface. So for example, if this is a cell, cilia would be present like this. Small hair-like protrusions, recesses. So they are present on the surface of a cell. Now so far, this is our cell. Yes, the nucleus hai. Let's draw maybe a mitochondria. The mitochondria aa gaya. This is its endoplasmic reticulum. Let's draw Golgi. Then there are ribosomes. All of this is a cell, and on the surface of the cell, what is present? Cilia are present. Okay. And can I say that this is basically my cell surface membrane? This is my cell surface membrane. So what happens is that basically this is our cell surface membrane. Okay. This is our cell membrane. What happens is that a centriole would go and it would attach near the surface of the cell surface membrane. So this is a centriole, and it would go and attach itself to the cell surface membrane. And now what would happen? The centriole would give rise to cilium. And how is it so? So basically, in a centriole, there were 27 microtubules present. There were a number of microtubules present, protofilaments present, right? So these filaments, they are chain-like, or they're like long chains, they're long cylinders. So what happens is that centriole would basically give rise to an exoneme of a cilia. We'll just understand what is an exoneme in a minute. Just hold on to this, but let me draw something. Okay. So basically, these are our long microtubules. Let's not go कि arrangement क्या है, किस तरह से organized है. Just notice that this is basically microtubules, and this cell membrane is now growing along this. So I can say that the cell membrane is covering or is enclosing these microtubules, right? This level, this was our cell membrane, okay? And this cell membrane, because the centriole has come and attached to the cell membrane, and now it's the microtubules, the filaments are now protruding beyond the boundary of that cell. The cell membrane is covering, the cell membrane is also being protruded, it is also being pushed along with these. So this is basically, this entire thing is what? This is a single cilium. So all the hair-like, thread-like structures that we see, the cilia, be it in animals, animal cells हमारे lungs में होते हैं, and they're present in our fallopian tubes. All these cells which contain these hair-like projections, these cilia are basically an extension of our cell membrane, and this consists of microtubules inside it. So basically, the region beneath the cell membrane, which is lying under the surface of cell membrane, this is termed as the basal body, which is the region below the cell membrane. That structure which is below the cell membrane, beneath the surface of the cell membrane, beneath the boundary, that is termed as the basal body. And the outgrowth, the thing that is outgrowing, the thing that is projecting out of the cell, and it is also being covered by the cell membrane, that outgrowth, that projection is known as the cilium. Is this clear so far, or do you want me to repeat this? I know this concept is a little new and a little difficult, so it takes a while to absorb it, but I'm trying to simplify as much as possible. Okay, perfect. Now let's understand, how does a cilium look like? We've seen the cross-section of a centriole, we've seen the cross-section of a microtubule. Let's take a cross-section, let's cut our cilia like this, and let's see how it looks like. So this is an exoneme. Exoneme is basically what we call the structure or the cross-section of the cilia. So if we cut open a cilia and we look at it under a microscope, it looks like this. ठीक है जी? Now, if you could notice, there is one complete circle, right? And then there is more than a semicircle, but this circle is not complete. I can say that one is a complete chain or a complete circle, and other is a half circle, right? So एक तो हमने ये चीज appreciate की that there is a doublet. This is basically a doublet. Two circles or two anythings, two species are coming together and they're forming a doublet. And one of them forms a complete circle and the other forms an almost semicircle, right? And let's count how many doublets do we have. 1, 2, 3, 4, 5, 6, 7, 8, and 9. So we have 9 doublets in the boundary region, okay? So we have our 9 doublets. And we also appreciated that each doublet has one complete circle and it has one semicircle, right? So basically these doublets are again microtubule doublets. हमने centriole में क्या पढ़ा था? That we had microtubule triplets. In cilium, we have microtubule doublets. And let's say this one, the one which is complete circle is A, and the one which is a semicircle, that is B. So we have 9 doublets of microtubules, and each doublet has an A microtubule which is basically a complete circle, a complete chain, and a B microtubule. ठीक है जी. Now basically this actually has 13 filaments, and this because it does not have enough space. If you could see, I can say that यहाँ पर मेरे पास 13 filaments complete हो रही हैं, और यहाँ पर it only has the capacity to have 11 filaments. So they're made up of the same thing. They do not have something different. It's just that one is made up of 13 filaments and the other is made up of 11 filaments. Okay? So this is a complete microtubule and this is a semi-complete microtubule. Okay? Now, let me just clear this diagram for you. So far we have appreciated that we have 9 microtubule doublets. One is a complete filament and the other is a semi-complete filament. And then there are 2 microtubule singlets. हमारे पास in the center we have 2 complete singlets. They are not incomplete, they are not semi-complete. There are 2 complete singlets, 2 complete microtubule singlets in the center. So first and foremost, let's understand the arrangement. It is a 9 plus 2 arrangement where there are 9 doublets in the surrounding, in the boundary of that structure, and we have 2 singlets in the center. So the arrangement of a centriole is a 9 plus 0 arrangement, and the arrangement of a cilium is a 9 plus 2 arrangement. So that is one difference between a centriole and a cilium. Then our centriole is present inside the cell, okay? And there is no membrane covering it. Cilium, on the contrary, is basically a protrusion of a basal body. A centriole is basically giving rise to a basal body. Centriole जाता है, cell membrane से attach हो जाता है. उसमें कुछ हमारे पास orientational arrangement changes आते हैं, because obviously centriole arrangement is 9 triplets and 0 hollow center. That centriole basically is attaching itself to the cell surface membrane. It is undergoing some modification. It becomes a basal body. Basal body is basically the region beneath the cell surface membrane, and all the filaments which are outgrowing outside of the cell surface, which are protruding, which are becoming those hair-like structures, those hair-like structures are called cilium, which are present outside the boundary of the cell, and they are also enclosed by the cell membrane. The cell membrane is growing along with them. There are 9 doublets present inside that structure, and there are 2 singlets present within the center. And now, every single A microfilament, which is an microtubule, sorry, every A microtubule, which has 13 filaments, it has this arm, these heads. If you could see, this looks like an antenna. हमारे पास ये कुछ ant-like structure बन रही है. If I could say, I can call this an ant basically. So every complete microtubule, every single complete microtubule which has 13 filaments, it has 2 arms on it, and they are basically dynein arms. Now, what is dynein? Dynein is basically a protein. Okay? Dynein is not just any protein, it is a motor protein. Now, just in a bit, I will explain what a motor protein is, but let's just focus on the structure first. So basically, every complete microtubule has dynein arms. And if you could see a pattern, I want to notice this pattern, that this complete microtubule with its dynein arms, these dynein arms are pointing towards the semi-complete microtubule, the semi-complete B microtubule. And subsequently for this one, these arms again are pointing towards the semi-complete microtubule, and these arms are also pointing towards the semi-complete microtubule. So this is basically the arrangement and the pattern. So there is one outer dynein arm, and then there's this inner dynein arm. तो जो इनर सेंटर की तरफ आर्म होता है उसको हम इनर आर्म बोलते हैं, और जो आउटर बाउंड्री की तरफ आर्म होता है उसको हम आउटर डाइनिन आर्म बोलते हैं. Now, what does this Dynein arm do? So you have studied in your O levels as well as in your A levels that cilia basically can move. They can move. They have a brush-like motion. They have a wave-like motion. They have a rhythmic motion. They can move. Now, how are they moving? What is basically the source or the reason why they're moving? So what happens is that these Dynein arms basically what do they do is that they can convert ATP, and we know that ATP is basically our energy currency. So they convert ATP, they convert the chemical energy of ATP into mechanical energy. Now, what do I mean by that? What happens is that these Dynein arms, ATP comes and attaches itself to the Dynein arms. ATP comes, it attaches to the Dynein arms, and what happens is that once it has ATP, it converts that chemical energy of that ATP into mechanical energy. And what it does is that these arms attach to the adjacent, to the neighboring microtubule, and then once it has attached, it contracts or it bends, which causes a sliding force, a sliding motion to be generated. Just give me a second. I will screen share this with you. Just give me a second. I'll show you a diagram and you'll be able to appreciate this more. I'm just screen sharing in a bit. Okay. So what happens is that we have these two doublets. This is one doublet and this is the other doublet. Now, if you could notice, one doublet is pointing to that end of the other doublet, which does not have those Dynein arms. So these Dynein arms, they're powered by ATP. And what they do is the Dynein arms of one microtubule doublet grip the adjacent doublet. जो उसके बराबर वाला doublet होता है, it once it has absorbed the energy of ATP, it then binds to the adjacent neighboring doublet, and then what happens is it pushes or it creates a force, a sliding force, and then what happens is it pushes it up, releases, and then grip it again. So a rhythmic movement, a continuous sliding force is generated by these Dynein arms. And why is it so? Because Dynein arms are basically motor proteins. They convert ATP's chemical energy into mechanical energy. And they generate a sliding motion, a sliding force, which enables that contraction, that movement of cilia. So any questions so far about this? Anything that you would unable to understand about cilia? Is the structure clear? Is the why is there motion? Everything great. Now, just like cilia, now when we talk about flagellum, and I said that sperm is one of those cells which completely moves by its flagella, its tail. When scientists basically saw a flagellum, now what's the difference between a flagellum and a cilia? So basically, this is a cell. This is a cell. Cilia are basically something like this, hair-like projections on a cell surface. And if I talk about flagella, flagella is something like this. It's a tail. Now, when scientists saw the cross-section of cilia, and we just discussed the cross-section and the structure and the arrangement of cilia, They observed the structure of a flagellum as well. And now over here, I'm talking about a eukaryotic flagellum. A flagellum in a protoctista, a flagellum of a fungi, a flagellum of any animal cells, case in point of a sperm cell. So any cell which is moving, which is locomoting, which is motile, and it is moving because of its flagellum, because of its tail-like protrusion, when they saw the structure, the cross-section of a eukaryotic flagellum, its arrangement was ditto same as this arrangement. So there is no difference in the structure, in the motion, in the function of a cilia and a eukaryotic flagellum. I'm not talking about a bacterial flagellum, strictly talking about a eukaryotic flagellum. The cilium and the eukaryotic flagellum both are basically protrusion of microtubules. ठीक है जी. They have the same arrangement, 9+2 arrangement होती है उनकी, Dynein arms होते हैं, which is causing the contraction, which is causing the motion, which is causing the movement. And both of these are present on the surface, on the boundary of the cell. और इसके भी इर्द गिर्द हमारे पास क्या होगी, ये भी हमारे पास enclosed by the cell membrane होगा. तो हमारे पास जो ये पूरा फ्लैजिलम है, ये जो आउटग्रोथ ऑफ माइक्रोट्यूब्यूल्स है, इस आउटग्रोथ को हमारी सेल मेम्ब्रेन इंक्लोज किए हुए होगी. व्हाई? बिकॉज इट इज बेसिकली द आउटग्रोथ. तो अंदर हमारी कोई बेसल बॉडी होगी. दिस इज आवर बेसल बॉडी and this entire species is our flagellum. And it again has a 9+2 arrangement where there are 9 microtubule doublets and 2 microtubule singlets. And that is about it. So any questions so far about the structure of microtubules, centrioles, cilium, flagella?